Hello everybody, welcome to the 6th lecture of Methods in Paleo-Environmental Reconstructions course. Today's topic is application of pollen analysis, as also is in the next lecture. So next two lectures we are going to go through the world of pollen analysis. The aims of the next two lectures, I hope after these two lectures you have some basic understanding of the principles behind pollen analysis as paleoecological tool. You also have learned some ways how fossil pollen data can be used to reconstruct past vegetation. And we will look at a few different ways how to do that. And then at the end of the second lecture, I will also give you a short introduction to non-pollen paleonomorphs as additional proxy for pollen. So these are the three main themes of these two next lectures. But what is then today's topic? So today's lecture is divided more or less into three parts. And first, I will give you a short introduction to pollen analysis. And then we will look a little bit how to present and visualize pollen data. And then also how to interpret pollen data. So basically we are going to go through the steps from how to obtain pollen data that we talked at the introductory lecture, like how to obtain paleo data. And then when we have, for example, a beet core, how to actually get the information of past vegetation out of this core. So basically through pollen identification and then how to interpret and present this data. So this is the main goal of this today's lecture. So first few words about pollen analysis. Basically, it means analysis of species and species relations interpreted from either recent or fossil pollen and spore records. During these lectures, we are, because this is looking into paleo environments, we are more concentrating on fossil pollen and spore records. And these can be used to reconstruct vegetation and also climate or other aspects in the environment based on what kind of pollen grains can be found from sediments. We can make interpretations what kind of environment has been at the study site during different times. If it's been more like open tundra-like vegetation or maybe coniferous forest or broadleaf forest, this can be detected from the pollen data from pollen analysis. Then related term to pollen analysis, which you can hear, is palynology. And basically it means the study of recent and fossil microscopic particles, including pollen and spores, but also other small fossil particles like fungi and so on. So this is just basically microscopic particles identified from the sediments. Then at the introductory lecture, I also introduced the term taphonomy, but because it has a big importance when interpreting the pollen record, it is good to keep in mind that taphonomy means the processes affecting the origin or the dispersal and deposition and preservation of the fossil remains of different fossil plant and animals. So basically, kind of the processes that affect then the fact how the pollen record will look like when it's analyzed from the sedimentary records. Then a few words about the principles of pollen analysis. So basically on which facts or factors is pollen analysis based on. First of all, plants produce huge amounts of pollen and spores, and as a reproductive purposes, and those spores and pollen grains are mixed in the atmosphere. So it will result in a homogeneous pollen rain that will then be deposited on ground or on land or water, and then preserved there. And usually pollen grains are best preserved in oxygen poor, so basically anoxic environments. And usually these are lake bottoms and peat layers that the pollen grains are usually obtained from. Then one important thing to keep in mind is that when we have the sedimentary pollen, the pollen from certain sediment layer, it reflects the surrounding vegetation in space and time. So basically it means that the pollen at the sediment record, it kind of reflects the vegetation that has been growing around the site at the time of the deposition. Also important factor is that pollen grains can be identified to different taxonomic levels. There is some species or some occasions that pollen can be identified to the species level, mostly to the family or genus level. But anyway, in the level that you can make interpretation how the ecosystem or the vegetation community has changed in the area. So then basically, when we look at the sediment sample and what is the pollen composition in this sediment sample from certain depth and from certain age, we can make reconstruction of the past vegetation in the area. Also, when we look the entire sediment core and analyze series of sediment samples throughout certain time series, we can look how the vegetation have changed through time. Or also if we'll take sites from different areas, or if we have, for example, a region, for example, we would want to research how vegetation has changed in southern Finland or from east to west. We can take different sites from this area and investigate the pollen through time from those sites and make this kind of interpretations about change in vegetation patterns or the vegetation pattern at certain time. So these points are the kind of the basic idea behind the pollen analysis and how it can be used to reconstruct past environments. Then a few words about the history of the pollen analysis. It is not a very old method, although first studies of pollen in sedimentary records were more common or started to be more common at the late 1800s, but already first observed fossil pollen in sedimentary rocks was done by Göppert by 1836. So these are the most earliest kind of pollen studies, but the actual start of the method as to reconstruct past vegetations comes down to Leonard von Post, and he is considered as the pioneer of the modern pollen analysis. And he was first of all a geologist, studying geology, also zoology in Uppsala, and he was professor in Stockholm. And he got interested about the pollen in the sediments, and also was the first to present pollen diagram, stratigraphical pollen diagram from a peat deposits. So basically, the start of the pollen analysis as a method is often traced back to his lecture in 1916 in Oslo, and there he presented his diagram of forest tree pollen in South Swedish peat bog deposit, and this is a rough kind of sketch from from the first pollen diagram that he presented. And here you can see the curves of different taxa and how they change through the different depths of the beat record. And this study was actually translated into English only as late as 1967. Few words about the development of the pollen analysis methods. So when it was introduced, there was quite rapid development in the method, especially in the method of preparing the samples through chemical treatment in the lab. There was, because the basic idea when you are preparing the pollen sample is that you take a small subsample and then you try to remove all other material except the pollen grains so that it will be more clear to count and identify the pollen from the sample under the microscope. So these methods were developing quite fast, and there came this hydrochloric acid treatment, which basically is used for removing mineral material from the samples. Then also acetolysis done with, for example, it's sulfur acid to remove more like cellulose and extra plant material. So basically there was a big developments in the method to get more or get better samples. Then also the kind of the introduction of or the interpretation of the pollen samples. There started to be interpretations or division between the forest cover and more open areas. So basically calculating sums for air-boreal pollen, which means more forested cover, and is pollen that is more transported through wind, or non-aerial pollen, which is more like grasses and flowering plants, which is more like reflecting open areas. So these were the kind of the first developments in the field. Then also one important step was the more precise development of the dating methods, especially in the radiocarbon dating, and that also then gave the opportunity to create more precise and chronologically better reconstructions of past vegetation. During the last decades, there has been this trend, and that the pollen analysis has gone more and more towards using from the qualitative analysis of how vegetation have changed more towards numerical analysis and using different statistical methods to look more into the changes or relations of different data or different factors affecting pollen data. Also, because of the development of the dating methods, there can be more high resolution analysis done and more precise chronology. Also more effort and time have been put into high resolution analysis so that the actual temporal resolution would be more closer to the modern ecology. Also, there's been developments in the spatial resolution of the analysis so that the paleoecological data and modern ecological data would be more comparable. So these are some of the developments. And the use of pollen analysis in environmental research, there's been more and more applications for this. And traditionally it's been used as a tool for reconstructing past vegetation. Now it's also more quantitative vegetation, trying to model the distribution of different species, also the biomass of different species. Also, pollen data can be used to reconstruct past climates, or ecosystem change, and even they have been lately used to reconstruct different disturbance events in the area. And these we are going to then look more next lectures. Then just briefly about the origin of pollen. As I said, plants produce huge amounts of pollen, and mainly in the purpose of the reproduction. But because the amount of pollen is huge and not all of the pollen will end up to the other plants and actually perform the as in the reproduction purposes, and most of it actually will fall to the land or water surface and is accumulated, and that is one of the reasons why we can use pollen analysis. And the amount that plants produce pollen, it differs from species to species, also from habitat to habitat. And there's different pollination strategies, which also affects the amount of produced pollen in different plants. And there can be wind pollinated, which usually are then transferred or transported through air, through wind, animal pollinated, and then self-pollinated. And when it's animal or self-pollinated, then there is usually less pollen produced by the species. And there's different kind of methods of the pollination between different species, between flowering plants, or for example in conifers. And as I said, the plants, they produce huge quantities of pollen. And for example, one anther of birch can contain over 7,000 grains, which means that the whole flower can have 6 million pollen grains. And that's quite a lot. Then as comparison, a flower of hemp can produce 70,000 grains. And then if we take one of the most common trees in Finland, for example, one branch of pine tree can produce 350 million pollen grains, which is a huge amount of pollen. And you can realize that then this will be enough to also have some of it deposited on the sedimentary records. And as I already said, there's a big difference between the wind pollinated and insect pollinated plants and the amount of pollen production. And it is also possible that even if we have same species, for example, a pine tree, in different climate or in different habitat, it can produce different amounts of pollen. For example, if it's open site where the tree can get more light and more nutrition and more preferable living conditions, it can produce more pollen than if we would have the same species in the dense forest where there is more competition with other species. So this can also affect to the production of pollen. And then also these differences and changes in pollen production can also then obviously affect how much of the pollen can be deposited in the sediments, and if it's actually maybe some points overrepresenting or underrepresenting the actual amount of the species in the landscape. Then a few words about pollen dispersal. Basically, pollen is everywhere. You can find it everywhere in the world, including continental ice sheets. So wind is transporting pollen through air and atmosphere, and it can be found even in the remotest parts of the globe. And usually the pollen is transported different ways, and well, as wind pollinated through air and so on. And that also can affect what kind of pollen accumulation is in the site. The different components of the transportation, you can have this kind of a component of that is called a trunk component, which means that you get pollen that is mostly from under the canopy of the forest, or you can have the canopy component, which comes over the tree canopy, which is mostly the wind transported, or the rain component, which comes down with the rain, which is also then pollen probably transported from, could be from the longer distances. Then there is this kind of inverse component, which, for example, to lakes and some mires that have their surface level lower than the surrounding environment, you can get pollen with runoff from the surrounding areas. Or then you can have this local component, which basically comes from the local vegetation. And this all is quite important for the interpretation of pollen data. And the size of the site is in quite big role in this. And in general, the bigger the diameter of the site is, the bigger the source area of the pollen is. And usually this source area can be divided to local and regionals. In 1981, Jacobson and Bradshaw, they made this kind of a schematic figure where they divided the source area of pollen in 3 classes, meaning that, for example, if you have site that is less than 100 meters in diameter, then over half of the, or almost 80% of the total pollen rain coming to the site is from the local or component or from the near of the study site. Whereas when you go to the kind of more bigger sites, is if you have, for example, lake that is a few hundred of meters in the diameter, then you get more this kind of long distance pollen, and the more or the bigger proportion of the pollen can represent more like regional vegetation in the surrounding area. So when interpreting the vegetation and pollen data, the size of the sampling site is very important to keep in mind. Then one reason why pollen analysis can be done is that the pollen grains actually preserve quite well in the sediments, and this is because the structure of the pollen, and it's exine, the outer layer is formed this kind of a strong material called sporopollenin, and it is said to be one of the hardest materials, and this enables the preservation of pollen grains. And this can be seen also because when preparing the pollen sample, as I mentioned, we use this hydrofluoric acid, which is used to remove the mineral material from the samples. So basically, when we have a sediment sample, and if we have there, for example, clay or other mineral material, the used chemicals melt down the mineral material, but then still pollen grains remains in the sample. So this shows that the pollen is quite strong in its structure. And usually the paleoecological proxies are best preserved in anoxic and acid environments, and usually studied from lakes or marine sediments, peat layers. And even if we had a short introduction to the main archives in the introductory lecture, I thought that I will just say a few words about the main sites or archives that can be used for obtaining pollen records. So basically these anoxic environments usually are lake bottoms or mires, and in lakes you get quite a lot regional pollen, also depending on the size of the lake. some of the local pollen, but the local vegetation is usually aquatic vegetation, and usually the aquatic plants are more self-pollinating and are not producing that much pollen, so it doesn't cause very big bias in the results. In lakes, there might be the situation that you get pollen with the runoff from the catchment, which then can need to be taken into account. Also, there might be some resuspension or redeposition in the lakes, but mostly the sedimentation rate is very stable, and usually the sample is taken in the middle part of the lake, which is the most peaceful sedimentation environment. So these are the main points for the lakes. But then if you want more local presentation of past vegetation, then you usually use, for example, something smaller than lakes. Then mires, if we talk about large open mires, usually peat sediments is easy to date because there's a lot of macrofossils that can be dated for radiocarbon dating, and it's quite sure that peat is not resuspended or there is not mixing, but then there can be often very variable sedimentation rate because the peat accumulation can change because of the climate. Sometimes there can be more humidification of the peat, or it can accumulate more fast in some kind of different climatical environment. So this can affect the record. Also, sometimes if it's very fresh or in this peat layers, the concentration of microfossils can be low, and it might make the samples more, it might take more time to identify and count the necessary amount of microfossils. And then opposite to lakes, mires can have quite strong signal from the local vegetation. You can see from here these pictures, you can get, for example, pine or some ericaceae like heath heather, or then, for example, you can have some other species on the sides. Here you can see there can be birch trees on the side that then can be overrepresented in the pollen diagram. Then if you want to study very local, small scale development of a forest, you can take sample inside from small forest hollow, which is basically also usually peat core. It's small peat depression in the forest. And the idea here is that the pollen is not transported from long distance, but it's more local and trunk component. So basically showing the changes in the vegetation within a few hundred of meters in the site. And these have been lately more common as the results from these local studies can be more comparable with the modern stand scale changes or studies in forest dynamics. So then this has been one development in the analysis as well. In the introductory lecture, we look at the steps of environmental research. So basically obtain the core, subsampling, then preparing the samples in the lab and the microscopy. And in pollen analysis, as I already said, there's these standard chemical treatments that can be used to remove the extra material from the pollen slides. And the aim is to obtain a microscopic slide that then can display different pollen grains, and which can be then identified and counted from the pollen slides. And as I said, pollen grains can be identified to family or genus, or even species level, because of they have different kind of structures. And the identification is based on 4 main morphological components, usually the shape and size of pollen grains. For example, there's different kind of amount of small holes, like pori, or this kind of a feature called colby, which is this kind of a striate, or how would I say, kind of a wound on the pollen surface. And also there are different species or taxa can have different amount of these structures, and also there can be change or differences in the shape and in the size. And for example, pine pollen have this kind of a structure in it similar to spruce, which is called this kind of a saccate. And usually pine and spruce are more bigger than, for example, birch and oak or willow, for example. And then also there can be different surface structures. Like here you can see there is this kind of a reticula on the surface on the willow pollen, when birch pollen has quite smooth surface. So these textures and these morphological structures can be used to identify different pollen grains. And in this course, we are not going to look more into identification, but as far as I know, there should be a pollen analysis course held by Professor Heikki Seppä in January and February, if it won't be cancelled, but I'm sure you will get more information about the course shortly. But in this course, there is going to be actual practice how to identify, and you will get to do field work and actually kind of complete a full study, beginning from field work, then preparing the sample, then doing the pollen identification under the microscope, and then interpreting the results. So this is more like a hands-on course, and if you are at all interested, I recommend the course. It's very much fun. So then when we have the actual pollen data, how do we then use it, and how do we visualize the pollen data? So next few words about what type of data there can be in the pollen derived from pollen record, and how we can visualize the data. So presenting pollen data, we can present the data in different kind of data sets. So basically just as numerical data sets, or most commonly pollen has been presented in this kind of a stratigraphical pollen diagrams, where the pollen data is projected through time or depth, but usually through time. And also then there can be maps presenting pollen data, for example, change in the pollen, how the pollen records show the change in vegetation cover in different times, or maps showing the migration or changes in distribution of different species. But these are the main ways of visualizing or representing data. There's different kind of data sets. So basically always when you count and identify pollen grains under the microscope, you get the so-called raw data, and usually that's in Excel or any other table format, usually, where it's then easily analyzed and visualized. And there can be 2 types of pollen data sets, relative pollen data and absolute pollen data. And basically what we mean with relative pollen data is data that gives percentage values compared to a certain pollen sum. So basically you get this kind of relative pollen diagrams, which we will look more closely shortly. And then there's absolute pollen data. And that means you can have the absolute concentrations or pollen accumulation rate, and those can be then absolute pollen diagrams. And then usually the pollen data is presented as a stratigraphic data, long time sequences, and those can be usually relative or absolute data. But then there is also, you can do this modern pollen analysis from the surface samples. For example, if you have to make this kind of studies how, for example, modern pollen reflect the actual modern vegetation, and usually this is then used as a relative data. And then recently, there's been more and more development in pollen databases, where basically researchers input their own data that they have produced and published, and then using these big databases can be done more large-scale studies of regional and global ecosystem change, and this has been quite big trend recently. And there's different kind of databases. I've put there a few links for paleoecological data, and here as one example, just how, for example, Neotoma database that has sites and pollen data and other fossil data also from all over the world. They have this kind of a map explorer that you can use. For example, you can search by taxon, or if you want certain time frame, and then you get the sites. For example, here it's from Europe, there are speech records that have, for example, Holocene record of beach. And this is then from these sites, you can download the data and then, or download, for example, data from certain area and then do a bigger study than if you would only have your own one site. Then about the relative and absolute pollen data. So basically the relative pollen data means the proportion of certain species of certain amount of pollen counted. And these percentage values do not actually reveal the abundance of the species. So it doesn't, for example, if we would have a pollen record that says that some species increases, for example, 30%, and other species proportion decrease about 10% or so, it doesn't necessarily mean that there's a big change in the biomass, but it can be that the change between the species is more relative. So basically, the relative values give more information about the composition of the forest rather than or the vegetation rather than the actual abundances of the species. And even the percentage values change the biomass. Usually the percentage values can stay very much the same even if there is change in the biomass. So then we need to consider what we actually want to study when we look at the different data. So here is example of the relative data. So basically we can have a situation where we would have a vegetation community that has less biomass. So for example, less trees. We can think that the black, red, and blue dots are presenting different tree species. And here we have 50% of the trees are red, 40% are black, and then 10% are blue. But then we can have a similar situation in other area where there is way more trees and bigger biomass, but still 50% of the species are these red ones, 40% these black ones, and 10% of blue. So when we look at the relative pollen diagram, you have to keep in mind that it presents the change in the species composition rather than the abundance. And then absolute pollen values is then when you actually can get the pollen concentration per, for example, square centimeter, or you can get the pollen accumulation rate. So basically, what is the amount of pollen accumulating on a certain area in 1 year? For example, how much pollen accumulates in 1 year in 1 square centimeter area. And the bigger the pollen accumulation rate is, then bigger the species abundance is interpreted to be. And for example, in tree lines, if you're studying change in the tree line and you are interested about the amount of the species rather than the change in the species composition, you can use bar values. And also, if we want to look at the change in the population growth, we can use bar values. And also, we want to look, for example, changes in the pollen production of certain species. We can look into the absolute pollen values, but then also there are some problems that can cause problems with the identification. It's always depending on the concentration is always depending on the amount of pollen or the sedimentation rate. So when using the absolute values, we need to have really good chronology for the course. So we need to have a stable sedimentation so that we can say that the accumulation is even and that the sedimentation rate doesn't affect the pollen accumulation. And then also, for example, it can be biased from the secondary deposition, or if there's some disturbance in the sedimentation, or if there's more runoff in the area, then there can be problems in the reliable absolute pollen values. But it's been used quite successfully. Then about pollen diagrams. So basically then when we have pollen data ready, then it's usually visualized and presented because it's easier to see the changes when you're looking the visual production rather than just the numbers. So this kind of stratigraphical diagrams are the traditional way of presenting pollen data, and both relative and absolute data can be presented in the similar diagrams. And there's different softwares making this kind of stratigraphical diagrams. There's this called Telia software by Eric Grimm, C2 program by Steve Juckins, and now that more and more everything is starting to be in R software. There's also this new RioJa package in R that can be used to do pollen diagrams. Because for example, Telia and C2, they have only limited access without a license fee, but then in R, everyone can use it and it's free. So it's a good and welcomed development. So then if we look more closely to the pollen diagram, what are the parts in it? There's first of all, there's the depth, and then there's the age. And as Jon probably told in your lecture, when we are looking at the past, trying to reconstruct the past environments and looking at the paleo data, the chronology is crucial and dating the sediment, because if we don't have the time frame or the chronology for our core or the data, it doesn't really say anything. If we wouldn't have the knowledge that, for example, here spruce appears to the vegetation around 3,000 years ago, if we wouldn't know when this happened, it wouldn't really say much. So the chronology is really important. Then there's sometimes this kind of a lithological information. So basically how the core changed, but this, I think nowadays it's starting to be more rare. Then here is actually the actual data. So the pollen curves, they can be presented as this kind of silhouette curves or bars or lines or however visualize it, but presenting it this kind of silhouette curves are the most common ways usually. Then usually at the top you have the name of the taxa. So basically here you have Alnus, Betula, Pinus, Picea. So basically different trees, and usually they can be grouped by different pollen groups. So it can be divided first, for example, trees, then shrubs, then herbs and grasses, and so on. Then sometimes the pollen diagram can be divided into different zones, and basically based on the changes in the data. But these are usually what you can see, the main elements. Then also usually it says that which side, if it's the relative or absolute diagram, this is percentages, so it's a relative from the whole sum. Then usually there's the analyst of the who's analyzed the pollen in the diagram, and so then just showing you the relative pollen diagrams and how actually we obtain the relative pollen data. So basically the relative proportion of changes in the record, as I already said, and the principle of creating this relative pollen data is that the percentage value of one species is counted from the so-called total pollen sum, which is usually marked with the P. And the total sum counted can vary depending what we are looking. And usually the most common amount, for example, if you are looking forest composition or vegetation composition, is general. From one pollen slides, you may target to count 300 to 500 pollen grains. But for example, if you're trying to look human indicators from the samples, and usually these are taxa that produce less pollen and are more rare in the pollen record. So then usually the sum counted is larger, and you can target to 700 or 1000 pollen grains. But if you go above this, it usually doesn't change the results. So basically how then the percentages are calculated, for example, in terrestrial plant communities, which is the most common what is reconstructed. So basically then the percentage values of each taxa is counted from the total sum of terrestrial pollen and spores. So basically the total sum would be trees, shrubs, herbs, and ferns taxa calculated all together. Like for example here, done this example. For example, here we have trees and some herbs and spores, and altogether the sum of the counted pollen is 300 pollen grains. And then from this 300, you count the percentage of each taxa. For example, 45 Alnus grains means that from this slice, if it's only this taxa, the percentage, the relative proportion of Alnus of the vegetation composition is 15%, and so on. So this is how it's done. And usually then aquatics and, for example, sphagnum, for example, in the mire, if you have a sphagnum, you can have that very much overrepresented, that then can cause bias to the relative amounts. So then usually you calculate own sums from aquatics and sphagnum and not include that to the terrestrial sum. And here is just example of relative pollen data. Basically here you can see changes in the vegetation, main pollen composition is in this site is from trees. Basically there's spruce, pine, alder, birch, and here you can see the change, the relative change when there's, for example, more pine and birch, there's less spruce, but then there's a change in the vegetation and the relative amount of spruce increases while, for example, birch decreases and so on. You can see the changes. But then this is just the composition. We might have the same amount of biomass, or there probably wouldn't be change in the biomass even if there's change in the proportions. Then the absolute pollen analysis. So basically, as I already said, gives the information about the absolute abundances in the plant taxa and how then the absolute pollen data is produced. This happens when preparing the pollen samples. We want to know the concentration of the pollen grains in the sediment or the amount of the sample that we analyze. So basically then we take volumetric sample, for example, 1 cubic centimeter or half cubic centimeter, and there is this special marker grain tablets that usually contains some grains that are not that typical in the pollen spectra on the site. These can be usually, for example, eucalyptus pollen grains or Lycopodium spores or so on. So basically, one tablet contains known amount of marker grains, and when this is added at the process of preparing the samples to the sample, we will know the amount of the marker grains, and the tablet is added at the beginning of the laboratory treatment, so that we know that also the marker grains goes through same chemical treatment as the actual fossil pollen in the sample. And then when we count the marker grains per counted pollen sample, we can use this information to calculate the concentration. And usually the pollen concentration, or usually it is calculated by counted amount of pollen plus then the known amount of added marker grains, counted pollen multiplied by the added marker grains, and then divided by counted marker grains. So then this will give the pollen concentration. And then furthermore, we can calculate the pollen accumulation rate. So basically meaning how many pollen grains is deposited on 1 square centimeter per year, for example. And then this can be calculated that we take the pollen concentration, then we have the deposition time, and then we can get the pollen accumulation rate. So this is how we are producing the absolute data. And then here is just a diagram showing absolute pollen data. So rather than changes in the species composition, we can actually see that, for example, there's here time, that there's a decrease in the biomass of birch and pine, while there's increase in the biomass of spruce, which also can be seen in the composition, but this kind of gives additional information that there's actually change in the biomass of different species in the area. And then just a comparison of this relative and absolute pollen data. So basically here is, for example, pine, and then example of birch from the same site. And here we can see the difference, like for example, if we look at the change in the percentage of pine, we can see that there's increase in the relative proportion of pine in the pollen record. At the same time, there is a decrease in the proportion of birch. So basically meaning that the relative proportion of pine is more bigger, or the proportion of pine is increasing. And then towards the late Holocene again decreasing. But then if we look at the concentration and the pollen accumulation rate, we see that actually in both the pine and birch, the biomass and the actually abundant, not the biomass, but the abundance of pollen accumulation, it's decreasing. So basically it means that even though there's compositional change and it seems that there's more, the proportion of pine is growing, it's still at the same time the actual amount of the abundance of the species is decreasing. So when using this both, we can get information or different information about the site and how the vegetation has changed. And here, for example, this is from the northern area, northern Finland. So basically this means that the vegetation changed towards more tundra-like vegetation and the amount of tree species decrease, and that's causing the decrease in the abundance of the taxa. Then about visualization of the pollen data. So basically pollen groups can be, the pollen diagram can be divided in different ways. You can use different kind of groups, grouping the pollen with first presenting trees, then maybe shrubs, then herbs. There can be also aquatics. Then usually there can be this kind of summary of showing the whole sum of trees, the proportion of trees or shrubs and herbs. And here we can see, for example, that the tree taxa covers almost 100% or 90% of the vegetation composition. So the area must have been forested. There's also then there are some times habit of putting similar curves closer together, which could be also trees and so on. But also there's sometimes the pollen is present in the order of appearance. So basically the first taxa in the first sample, the taxa present are coming first in the diagram, and then you can see the change that from left to right, the amount of the type of pollen increases when we go towards the younger samples. Also, some people present the pollen in alphabetical order or in random order. Myself, I usually use the order that first trees and usually even that first conifers and then broadleaves and then going to shrubs and then to herbs and so on. But there's different ways to do this. Then pollen diagrams can be visualized by dividing it to different zones based on the composition of different samples. And this can be done visually or numerically, and it's kind of this is quite good example showing quite clear changes in the pollen diagram. So basically you can see here that this part, there's different or it's kind of a similar pollen composition, and then there's a change where you can see that there's, for example, big increase in proportion of pine and then decrease here in Artemisia or in Cyperaceae. And here you can see the division of the pollen zone. And then again, when there is a clear change in the pollen reflecting the vegetation, there's other zonation, and again, when there's a change, another. And this zonation can be done by just visual inspection, but it's recommended and it's more objective and clear and more repeatable if it's done by numerical methods, and usually this can be done using different clustering or ordination methods that divide the pollen samples in different clusters, and basically making the zone, the idea is that inside the zone the pollen samples are more similar than in the next zone. So this is done to help kind of interpret the changes. Then one way of presenting pollen is different pollen maps. So basically, it is this kind of spatial representation of pollen data. And I've just took here a few pollen maps. And for example, this map is from the same study that was the example of the pollen accumulation rates and percentage values of pine and birch. But here, the pollen data have been used to reconstruct this kind of a time window from 7,000 years ago, showing the vegetation in Finnish Lapland. So basically here at the upper map, we can see the vegetation, modern vegetation, showing that we have birch forest and then Arctic alpine tundra-like vegetation, and then here some birch, sparge, pine, and more like a little bit also birch, spruce, pine forest showing here. But then when we look 7,000 years ago, when it was the Holocene warm period, the tree and the vegetation zones have been in more northern location. So here we can see that actually now where we have just birch forest, there's birch forest with pine, and actually pine forest coming quite to the quite north with then some sparse birch. So there's been clear change in the vegetation, and based on the pollen record at this time point, this kind of map, they have been able to create this kind of map. Then this is just other example. This is also based on pollen records done from different locations from the globe, and this is reconstructing the vegetation during the last glacial maximum. Basically the gray area showing the ice sheets, and then different colors representing different kind of biomes, basically interpreted from the pollen records. And here we see that in Eurasia, there's been quite dry grassland vegetation, some temperate forest, some sites with boreal forest, mostly here, North Tundra-like vegetation. Then in North America, there's been in the east coast or eastern part more forested areas, more boreal temperate forest, and then more like savanna-like grassland vegetation, and then here more tundra. So basically using the pollen record, they derive this information and then presenting it in the map. Then as a third example, here's just map basically presenting the modern amount of volume of spruce at the moment in Fennoscandia. And then these lines represent the spread of spruce through Fennoscandia from the east. So basically pollen records from the transect from east to west, showing when the spruce appear in the pollen, and based on that they've done interpretation of the migration of spruce through Fennoscandia. So, and then this is presented on the map. It could be also presented in several pollen diagrams, but then this is one way of visualizing it. Then at the end, a few words about the interpretation of pollen data. What do we actually see from pollen data? So we basically see the vegetation composition in the research area, or the proportion of abundances of certain species. Then in the pollen diagram, when kind of creating the pollen diagram, we have to keep in mind, and when interpreting, that some taxa is rare in the pollen record, and usually the absence from the pollen record doesn't necessarily mean that it didn't grow in the area, based in the case of the rare taxa. And some of the pollen that can be found from the record may not be local. It might be transported from the longer distance, depending of the site. Also, the level of identification, if we can get to species level, or if it's more family or genus level, and what can we actually say based on this. So what then actually affects pollen data? There can be several factors, from the fieldwork and sampling to the laboratory, through the laboratory preparation of the sample, and then to the identification. And all of these can affect the actual or the final record that we derive from the sediments. Important point is the reliability of the data. Also that the core is obtained properly. We have good chronology, and there hasn't been any contamination from for example, if you sample during the summer, you can have a lot of contamination from the air in the pollen. So usually in autumn or early spring or winter is the best times to avoid the contamination. And then through the analysis and the identification, so that actually the pollen sample is counted in orderly manner. Also that all grains in the sample are identified or marked unidentified, and not just the ones that you know immediately. And then also quality of the sample. Also sometimes the chemical treatment may destroy some of the pollen, or then cause some kind of bias. And then also that the same sample, from the same sample you can do this kind of repeatable counts and still get the similar proportions of the pollen. Then also the number of counted grains can cause bias. The more that you count, the more different taxa you can found up to a certain point. So also this amount of counted grains need to be taken into account, especially, for example, if you are interpreting the diversity from the pollen diagrams. Then also making the interpretations of the change in the ecosystems or in the vegetation. It depends of the time resolution of the data. In fine resolution analysis, if you do, for example, every half centimeter or every second millimeter, you can get very good time resolution, but this is not usually the case. You are not usually able to do this high resolution analysis, and usually then the time resolution is coarser, and usually it can be tens of years or even hundreds of years, and then this can need to be taken into account when interpreting the data. For example, then it's good to take into account how, for example, the count of when we are analyzing the samples through the core, that from every slide and every depth you count the same amount of pollen types. For example, you won't get reliable results if at the bottom of the core you, for example, count 500 grains per sample, and at the top 1,000 grains per sample, but you have to have a constant number of the counts, and then you can actually make interpretation that, for example, if you have the constant count of the pollen grains, you see that the number of the taxa increases, for example, towards the present, and then you can see reliably the proportions change in the pollen. Then there's the representativeness of the pollen. Basically, as already said, the pollen percentages do not directly reflect the plant cover percentages. There's also changes in the pollen productivity. Some species might be overrepresented, some species underrepresented. So basically this means that, for example, some species have less pollen, but then it can be more represented in the actual vegetation. Also, usually the wind pollinated trees can be overrepresented in the pollen record, and also the pollen production can vary between the different habitats. And also the dispersal and depositional environment can cause changes in the pollen representation. You can have different amounts of pollen in peat or in lake. And there's many different ways to correct errors in pollen representativeness. There's different kind of models to actually model the actual vegetation cover, and some of these ways we are going to look in the next lecture. Then important for the interpretation is the scale of the study. As I already said, the dispersal or the transportation of pollen depends on the size of the site and also the characteristics of the site. And depending of the research questions that we have, and if we want to look regional or if we want to look local vegetation, then that is the grounds for choosing the site and what kind of, do we want to sample lake sediments, or do we want to sample mire or this kind of a small forest hollows. And the basic scale of dispersal of pollen to the site, for example, from small study site, we can be sure, and these forest hollows, that they reflect very local vegetation, especially if it's very small site, then it can be just the surrounding forest or plant community that is represented. And if we want to look, that's very local. Or if we want to look for a large scale regional vegetation, then, or in this kind of a landscape ecological level, then we can go to a bigger lake where the pollen is transported from with the wind. For example, if you look at these photos, we have quite large open lake here where we can imagine that pollen comes with the wind and rain and then deposits at the lake sediment layers. Then we have here at the lower picture this small forest hollow which is inside the forest, and most likely the pollen comes from the surrounding trees and the surrounding vegetation, giving a different information between these sites. Basically the area that the pollen that have meaningful effect on the pollen record, it's called relevant source area of pollen. And basically the relevant source area of pollen means that the size of the area that is around a given study site, beyond which the distance, the correlation between pollen and the surrounding vegetation does not improve. So basically with the small site, you have a smaller source area. With the larger site, you have a larger source area. And this is based on this mathematical calculations that is called Prentice-Sugita model. And they've developed this kind of a dispersal and defining the source area, and the relevant source area is very central concept in this model. And this is some example of the effect of the size of the sample site. So basically in this study, they've studied 30 different sites with the radius of 2 meters, 50 meters as the relevant source area. And you can see that in smaller sites, when we look at the pollen proportions of different species, when we have sites with the radius of 2 meters or 50 meters, we get more variation between the sites. But then when we have a radius of 250 meters or even 750 meters, there's not as much variation. So here we can see that here we may get more regional change, but we don't see that big local changes in the pollen record. Then there is also numerical methods used in pollen analysis, because you don't always see the all the information, or it's difficult to get all the information from pollen diagrams. So usually pollen data is analyzed by information that has that comes from statistical methods or different numerical analyses. And it's more and more commonly used because nowadays it's the pollen analysis as a method, and in environmental reconstructions is more used to this quantitative analysis and so on. And also when we are using numerical methods, it also simplifies the data and maybe shows some certain structures or directions in the data and how it changed. And usually most commonly, for example, the cluster analysis that is used to make the zonation, or then different ordination methods. Also, there's different kind of more and more methods that are used also in modern ecology, is also used in pollen analysis, especially looking at the chains and relations of the different pollen data, or for example, different drivers of the change in the pollen record. And these include, for example, variation partitioning, time series analysis, different vegetation models, and so on. And you will hear more about these different statistical methods and how to analyze failure data statistically in Jon's lecture on Monday, 23rd of November. And therefore, I'm not going more into this, but you will learn more then. So as a summary for today, pollen analysis that is based on the great amount of pollen that is produced, dispersed, deposited, and preserved in lakes or beads or other sedimentary environments. Also, I want you to remember that there's different types of pollen data. There's relative pollen data and absolute pollen data that tells a different type of information of the past vegetation. Also, there's many factors affecting the pollen records that affect also the information that we are able to get from the records, and also what we see from the pollen data and from pollen diagram, and how to interpret the data. And recently there's a huge increase in the use of numerical and quantitative methods in vegetation reconstructions. And in the next lecture we will have we will concentrate more on this. But for this lecture, there will be the online tutorial on Wednesday at 2 o'clock. And there I will give you an assignment related to this lecture, and you have possibility to ask questions relating to this lecture and this topic, and we can discuss it further. And then the next lecture, we will look more into the ways to actually use pollen data to reconstructing vegetation, for example, migration patterns, how we can study diversity change through pollen. And then also we are going to see some short introduction to non-pollen palynomorphs. So this was all in this lecture. I thank you for your interest, and I hope that I see many of you on Wednesday at the online tutorial, and we can have some discussion about this lecture, or if you have any questions, you are welcome to ask on the online session. So thank you, and see you later.